Next one is the development of anther and formation of microspore or pollen grain. The young anther consists of homogeneous mass of parenchymatous cells surrounded by epidermis. It soon becomes four-lobed. In each of the four lobes, some of the hypodermal cells begin to act as archisporial initial. Each archisporial initial divides into an outer primary parietal cell and an inner primary sporogenous cells. The primary parietal cell divide to form 3 to 4 layers, that is endothecium, middle layer and tapetum. The primary sporogenous cells divide to produce a mass of sporogenous cells or microsporocytes. Each microspore mother cell divides meiotically to form 4 haploid microspore or pollen grains and remain arranged in tetrads. The arrangement in the tetrads can be tetrahedral, isobilateral, linear, and T-shaped and dicassate. Now the microspores are separated from tetrad. Next one is the development of male gametophyte, that is the microgametogenesis. The microspore or pollen grain is the first cell of the male gametophyte. It is unicellular and haploid. The shape varies from oval to polyhedral. The wall of the pollen grain is made up of two layers. The outer layer is exine and the inner layer is intine. At certain places, exine remain unthickened or missing, and these places are known as germ pore. So this is the different stages of microsporogenesis under the microscope. If you see the slide under the microscope, then you can observe different stages of microsporogenesis, that is the formation of pollen grain from pollen mother cell through meiosis. Here you should know one more point, that microspore start germinating in situ, that is while enclosed inside the microsporangium or pollen sac, and is called precoccus. Microspore may be best defined as partially developed male gametophyte. Microspore nucleus divides meiotically to form a smaller generative cell lying next to the spore wall and a much layer vegetative cell, which is also known as tube cell. A callose layer is deposited around the generative cell. The generative cell loses its contact with the wall of the microspore and become free in the cytoplasm. The callose layer then dissolves. The pollen grains are shed from the anther at this bicell stage.